and welcome to Money Matters. My name is Charlie Huntington, and I'm Public Relations Chairperson for Life Sciences PA. Life Sciences PA is the voice of advancement for Pennsylvania's bioscience, medical device, CRO, and healthcare IT industries. Today, we continue our special series on science leaders in our local community. But before we get started, I'd like to remind our viewers that from time to time, financial issues relating to life sciences, healthcare, or technology matters or companies may be discussed on the show. These discussions are not and should not be viewed as financial advice. Moreover, since this program is pre-recorded and shown at a later date, please keep in mind that the information may no longer be current. You should always check with your financial advisor before entering into any financial transaction. I'm pleased to have with me as my co-host, Kimon Hatza. Kimon is a business attorney and partner at GrowthCounsel, LLC in Philadelphia, Pennsylvania. His focus is in the life sciences, healthcare, and information technology areas. Welcome, Kimon.
Charlie, thank you. Happy to be here.
Kimon, can you tell us what's new with your practice since our last show and maybe elaborate with any challenges you've seen about financing these days?
Well, a lot going on. Things have been busy, and we've had a number of M&A deals going on, not necessarily in the life sciences, but more in the tech space. We've also had a number of financing transactions, again, across industries, not particularly focused in life sciences, but with across industries, but a lot with a tech focus. We're growing. Practice has been busy, so we're actually adding an attorney. That's new. Attorney will be starting shortly, and she'll be operating out of New York. So we'll have somebody in New York. So that's we're going up from 5 to 6, Charlie. So we're growing. In terms of financing, that's a tough question to distill. There's a lot of things that could be challenging in a financing, and it really depends on what stage you're at. You know, if you're at an early stage, you know, it's really finding people beyond your friends and family, finding people who are willing to take a risk, and then what kind of an instrument are they going to invest in? Is it going to be a convertible note? Is it going to be a safe? And what's going to be the trigger, you know, for that conversion? You know, the follow-on investment. What's the amount that you're looking for that will trigger a conversion of that note into whatever the security is that you sold in the follow-on investment? It really is lots of times, depending on what the, you know, what it is, what the product is that you're trying to, you know, generate, obviously long term. You know, sometimes they just sell themselves and people are coming out of the woodwork. Other times it's really a tough sell. So, but there are challenges at all of the stages, and it's hard for me to you know, distill it down in a short answer, since I know we have to move on with a really terrific guest we have this evening.
Thank you, Kimon. Appreciate it. Congrats on the growth of your practice, and I wish your new colleague in New York all the success in the world.
Thank you, Charlie. Appreciate it.
We have a great guest today, but before we get started, I want to remind our viewers, here's how you can have your question answered on a future show.
You can have your questions answered on Money Matters. Please go to our website, money-matterstv.com. On our homepage, click on the banner on the right that says, Send us your questions. While you're on our website, you can find information about our host and guests, as well as show notes and links about this show and past shows. Money Matters is also available as a podcast on iTunes and Stitcher, so you can listen to Money Matters while you're on the go. That website address, again, is money, M-O-N-E-Y, -matters, M-A-T-T-E-R-S, TV.com.
It's with great pleasure that I introduced Dr. Raj Rajasekaran. Dr. Rajasekaran is founder, president, and CEO of Therapy Architects, LLC, and its subsidiary company, Allcron Pharma, which is focused on developing targeted cancer therapeutics for adults and children. Currently, it is developing a leukemia drug for advanced cases of chronic myeloid leukemia. He was the founding Cancer Center Director at AI DuPont Hospital for Children and was senior scientist at Helen F. Graham Cancer Center in Wilmington, Delaware. Before that, he was a professor and taught at UCLA, where he mentored many students. That's also where he received his MBA. He has a bachelor's and a master's in botany from the Presidency College in Madras, India, and a PhD in biochemistry from the Indian Institute of Science in Bangalore. He's done postdoc studies at NYU Medical Center and the Weill Cornell Graduate School of Medical Sciences. Dr. Rajasekaran is most proud of founding the Cancer Vision Foundation, whose mission is to provide practical and factual information about all aspects of cancer with lectures uniquely designed for students ranging from elementary school to high school globally. He's married, and he and his wife have 2 children. Raj, welcome to the show.
Thank you for having me.
Raj, what is the inspiration behind your work on cancer?
Yeah, it starts a while ago when I was a kid. The inspiration is from a profound childhood experience. You know, my father was a male nurse in a hospital, in a cancer hospital, in the government cancer hospital, actually. So I used to accompany him during school holidays and, to the hospital. So most often I see cancer patients lying still on the bed, covered with white cloth all over. So I used to ask my dad, so why are they covered like that? So my dad used to tell me they passed away because of cancer. He also said that when the cancer spreads throughout the body, it's incurable, and ultimately the patient dies.
Wow.
So these words stayed with me, and leaving a deep impression in me to start working and understand about cancer. So that's how I started, my work on cancer.
So when and how did you start to work on cancer?
So about 40 years ago, working on cancer in India, it's almost impossible. It's very, very challenging. You know, most of the people don't even talk about cancer, and cancer was like a mystery disease. And only when somebody is affected in the family, they realize about cancer. So one of the vivid memories in my mind, even today, is that because my father was working in the cancer hospital. A lot of people from the village, when they get cancer, they come to the hospital where my father works. And during their treatment, they stay at my house. And one such patient had terminal leukemia. And I remember about that patient, she almost lost all her weight. She cannot even get up. And her brother used to carry her for her basic routines. And then after a few days, she went into coma and passed away. But the thing is, my parents never told us what was a problem with that lady or with that patient. And that is usual with also all the other adults in the community. They never talk about cancer, or they would never tell to kids or any, they won't talk themselves about cancer because they thought it was so distressing to talk about cancer. So I was at that time thinking how I'm going to help these patients. So I thought one way is to become a doctor and help these patients. So in those days, the medical colleges were run by the government, and the admissions to the medical college was purely based on caste basis. It's not based on any merit or anything. So it was very difficult, and people belonging to the upper caste it's very, very difficult to get into, professional colleges. So I could not, after 3 attempts, I was unable to get into medical school, although I had all the qualifications. So finally, I decided to study botany, and which I really enjoyed. So during my studies in botany, I came across to work with a professor called T.R. Govindachari, who was considered as the father of natural chemistry in India. So he was working on a plant called Vinca rosea, which we all have in our backyard. And the roots of this plant contain an alkaloid, which is an anti-cancer drug. Even today, it is used as a drug for treating cancer. So I started to work with him on the purification of this compound in his lab. And that was the first time I ever got an opportunity to work on cancer. So during the time, I realized research and not medicine would be the path for me to find a cure for cancer. So looking back, not getting into medical school was really a blessing in disguise, and it redirected me to my lifelong commitment to cancer research.
Amazing story, Raj. So when did you move from India to the U.S. and start working on cancer?
Yeah, so as I mentioned, I did my botany, I did my BS and MS in botany. So one of the most exciting thing I learned during that time was one of the characteristics of the plant cells. Like now we know we have stem cells, it can even produce an entire person if you take a stem cells and do the work on that. But plant cells during that time, we know that each and every plant cell can produce another plant. That was very exciting to me. So I wanted to really test that myself and find out if this is really true, because it was like amazing to me. So I talked to my professors and they saw my interest, and 2 professors, especially Dr. T.T. Pandian and Dr. Govindarajalu, recommended me to do research at Madras University, the Center for Advanced Study in Botany in Madras University. So there I had the opportunity to work with Dr. Bala Subramaniam, we call him Dr. Balu, on plant tissue culture. So that was it's a great opportunity for me. And Dr. Balu, recognizing my interest, gave me full access to his lab. And I really enjoyed all my extra time doing research in the lab. So seeing my interest and commitment for cancer, Dr. Balu suggested, Raj, you should go to Indian Institute of Science for doing your PhD. And Indian Institute of Science at the time, even now, is considered as the Harvard University of India. To get in there, it's very competitive, and only few students every year are selected for PhD in that institute. So, and the selection is purely based on merit. So I thought I will have a chance. So I was admitted into the Indian Institute of Science for my PhD in biochemistry. And I was working on the biochemical adaptation of thermophilic fungi, which grows at high temperatures. So I learned a lot about biochemistry. It was incredible. You can't believe how a cell is able to control so many biochemical reactions in each and every second of its life. But the biochemical reactions from fungi to all the way up to human, they're all conserved. They are all the same. So that gave me a very broad vision about the cell and and its associated functions. So at that time, after finishing my PhD, it's very difficult from a botany background to go on to cancer research, because people will say, hey, you don't have the right background to do cancer research. So that happened to me many times. So finally, I landed up in doing a postdoctoral fellowship in cardiomyocytes and atrial natriuretic factor. So during the time, I realized I really have to study cell biology to understand know deeply about cancer. So I was looking for an opportunity to study cell biology, and I was fortunate to get into one of the most famous cell biology labs in the country at NYU Medical Center, where I studied how the proteins are trafficked within the cell. So during the time, I understood that 95% of the human cancers are derived from epithelial cells. And these cells that cover the body surface and also line the body cavity, like the esophagus, the intestine, the stomach, kidney, all are lined by these epithelial cells. So I decided to learn more about epithelial cells. So I did another postdoctoral fellowship with Dr. Enrique Rodriguez Bolon at Cornell University Medical College. So, and during that time, I came across a professor and chairman of the urologic oncology department, Dr. Neil Bander, who had who was very famous for his kidney cancer research and treatment. So I told him about my interest in cancer research and cancer metastasis, and he was very kind enough to offer, Raj, I have hundreds of kidney cancer tissues in my freezer. You're most welcome to use it and try to work and whatever you want to do on cancer. So that was the whole beginning of my career, where I started working on real cancer tissues, and I started to work on kidney as my model system.
Raj, how would you explain your research, your latest research, I guess, in lay terms to our viewers?
Yeah. So my research centers around understanding cancer metastasis and how the ways to stop it. So epithelial cells, as I said, they are the cells. 95% of the cancers originate from the epithelial cells. These epithelial cells are tightly attached to one another, and this attachment is because of a protein called cell adhesion molecule. The cell adhesion molecules are like molecular glue. They keep the cells tightly bound together. And when the cell becomes cancerous, this molecular glue disappears or become non-functional. So then the cells have the opportunity to leave the normal tissue or the healthy organ and then move into different parts of the body. So that's how you have cancer metastasis. So this loss of this molecular glue is one of the initial steps in cancer metastasis. So I decided to study that particular steps. So I started studying cell adhesion molecule, first. And so that is a very huge field. And the cell adhesion molecule in many organs have already been well characterized. But the thing about kidney, it was not well known what is the real cell adhesion molecule in the kidney. But one of the functions we all know about the kidney is to remove toxins from the blood. It takes out the salt we take, is all constantly excreted from the kidney into the bladder and to the urine. So there is a protein that regulates this salt levels in the blood, and that protein is called the sodium pump. So this protein is very abundant in kidney. So when we looked at the cancer tissues, we found this protein almost vanished in all the cancer tissues. So that gave me the clue, probably in kidney, this is one of the cell adhesion or the molecular glue. So we started looking into that idea, whether this sodium pump protein is a molecular glue in the kidney. And in fact, we verified and confirmed that is a cell adhesion molecule in kidney. And then when we reintroduced this molecule into the cancer cells, we also saw that the cancer cells were becoming like normal cells, unable to move, and they were all sticking together. And then eventually we tried to understand how this molecule is lost in the cancer cells. So once we understood that mechanism, that enabled us to develop some drugs to target to increase the level of this protein within the cancer cells. So that's how I got into drug development studies. So we did a lot of research and a lot of drug screening, and finally identified few drugs that can increase the level of this protein in the cancer cells in kidney. So some of those drugs are even used today in the treatment of metastatic kidney cancer. So after that, so these drugs, as you know, the drug development is a very complicated process. It needs a lot of work, a lot of money, and all those things. So all this work I just mentioned within this last 2-3 minutes, I have to tell you, it took almost 15 years of work of many dedicated scientists. So those scientists, their hard work has really paved the pathway for my career into stopping cancer metastasis research.
How did you transition to starting your work on childhood cancer?
Oh, that was a good question. So after I finished my tenure at UCLA, I had an opportunity to lead a cancer center at the Alfred I. DuPont Hospital for Children. So there it was a very challenging position because I have to start everything from the scratch, including the construction of a building and making facilities for cancer research, especially childhood cancer research. So that gave me a lot of time to study about childhood cancer. And so I understood that because the childhood cancer is rare compared to adult cancers, there are not too many new drugs in the new drugs for treating childhood cancer. Children are being treated with the same old drugs. And because of these drugs, the kids have very severe side effects, and some of these side effects they have to face lifelong. So that was one of the major problems in childhood cancer treatment. So I decided, okay, we have to have new drugs for the treatment of childhood cancer. And another thing that I learned is that the cancer itself is not a problem for the kid, but mainly the treatment was a main problem. All the issues the kid developed is because of the treatment. So we need to have better treatment. So during the time, nanotechnology was an emerging field for the treatment of for cancer treatment. So I saw a lot of potential in nanotechnology in the treatment of childhood leukemia or childhood cancer. So nanotechnology is basically using nanoparticles. Nanoparticles are tiny particles. You cannot even see them with your eyes. You need an electron microscope to see a particle. And these particles are hollow, and there is space inside, and we can load the drug into that space and deliver the drugs to cancer cells. So once you load the particles with the cancer treatment drug, you can then include some sensors on the surface of the nanoparticle. So these sensors will look for cancer cells in the body, and once they find the cancer cell, they will bind to it and then deliver the drug to the cancer cells. So we decided to do this technology for childhood leukemia, and in fact, we took commonly used drug in the treatment of childhood leukemia, encapsulated in nanoparticles and delivered the drug, and showed the proof of principle. This would be a novel way to treat children to prevent or to avoid side effects for the future.
That's great.
So, yeah, that's how it started.
One last question about this. How have you financed Allcron Pharma, which owns as I understand, owns the IP for the leukemia drug that you're working on?
Yeah, so far the financing is from family and friends. Like you mentioned, early stage companies always have difficulties to raise money. So, I have personally invested a lot of time and financial resources, foregoing other opportunities to dedicate myself to this endeavor. So getting additional funds at this stage is a significant challenge. And so, like you said, when I go for funding, either from government agencies or from private agencies or from investors, they always say that You are at the very early stage. Come back when you have more data.
Mhm.
But getting more data, you need more money. It's like
Yeah.
chicken and the egg story.
It
It's
sure is.
very difficult to get. So we at least need another $5 million to take this to file an IND and start the human clinical trials.
So, Raj, what would, and we've been given sort of a 5-minute warning a minute ago, so I've got two questions for you. What would success look like in 5 years, let's say? And we've just been given the 3-minute warning now.
Oh, okay. So if we get the money and if we are successful, we can collaborate with a big pharma and do the further studies on clinical trials, or we can establish ourselves all the necessary things needed to start clinical trial and establish a huge company by ourselves. So both are possible.
Okay. All right. Finally, in a minute, can you explain, tell us in 1 minute about your favorite pastime, which is your love for bonsai trees?
Oh yeah, that's a great question, Charlie. I love to grow bonsai because it's an incredible thing. I have to tell you, I have an old pomegranate tree which is now fruiting. It is almost 16 years old. The
Wow.
bonsai trees gives you an opportunity to learn patience, perseverance, and to remain hopeful. That's what it is.
Oh my gosh. Wow. And a lot of that is helpful when you're running a drug therapy company, I'm sure.
Absolutely, it's very similar.
So how many years have you grown bonsai trees for?
I started growing bonsai trees when I was in my BS, 40 years ago. So when I left India, I had about 20-year-old trees. I gave it to my friends and families. But now I have trees which are about 20, 25 years old in my house.
I understand you're an author. You've written book. You're an author, many publications within the scientific community, but specifically you wrote a book on bonsai trees. Can you give our viewers, I'm sure they can find it, but give our viewers the name of your book.
It's called The Art of Bonsai. That is that's a technique developed by myself in my home, and that is to grow tropical trees as bonsai, not as the Japanese trees where it's very common. And unfortunately, those books are all sold out. After I finished it, I have to write a second edition to have those copies available.
Well, I may go out on eBay before Kimon does to look for try to outbid him. So, Raj, you have been a wonderful guest. Kimon, thank you for co-hosting, and congrats again on the success of your business. Raj, we wish you a lot of success. I want to remind our viewers that our next guest is Steve Kent from Financial Literacy. That's what he's going to be talking about. We wish you all a great holiday season, and thank you for joining us.